My name is Aaron Mertz. I am a 2016 Arnold O. Beckman postdoctoral fellow and carried out my research at the Rockefeller University in New York City while I was supported by the Arnold and Mabel Beckman Foundation. Dr. Beckman was the founder of Beckman Instruments Incorporated. He invented many devices that revolutionized the fields of chemistry and biology. Among his many inventions are the pH meter and spectrophotometer. He also gave rise to Silicon Valley through his funding of the first transistor company. In his retirement, he and his wife Mabel became significant philanthropists who supported a number of academic and scientific institutions. Perhaps the most famous quotation of Dr. Beckman's is, "There is no satisfactory substitute for excellence." This philosophy was a guiding force in his life and shaped his company and his career, and I'm so grateful to have carried out my postdoctoral research with that philosophy. Because of his generosity and belief in supporting young scientists, I was able to carry out my research. I also wish to acknowledge my colleagues at the Rockefeller University, where I was advised by Dr. Elaine Fuchs in her Laboratory of Mammalian Cell Biology and Development. Dr. Fuchs is a world-renowned biologist, and I greatly benefited from her mentorship and from my interactions and collaborations with my many colleagues in her laboratory. My postdoctoral research was about a class of tissues called squamous epithelia, which consist of multiple layers of cells and serve as protective barriers or cases of organs or of the entire organism itself. Specifically, I sought to understand how these tissues stratify into multiple layers. This process of stratification is essential to the development of many tissue types, including cornea, oral tissues, esophagus, and epidermis, which are characterized by layers of cells atop a basal layer, which sits on a basement membrane. These stratified tissues are distinct from non-stratified epithelia, such as the gut. The epidermis follows a well-defined stratification and differentiation program. In the laboratory, we used mouse embryos to study skin development. Mice have a gestation period of 19 days, and the skin stratifies between days 9.5 and 14.5. By day 18.5, shortly before birth, the skin on a mouse has all of its layers defined. This process is essential to the tissue's ability to serve as a barrier that keeps microbes out and fluids in. When the process goes awry, blistering skin disorders can occur. Additionally, some of the paradigms that underlie embryonic growth are represented in the growth of certain cancers. The major aim of my research was to determine how basal and suprabasal layers expand with respect to each other as epidermis grows and develops. I explored how geometry and mechanics influence early epidermal stratification, how epidermal divisions contribute to development of epidermal appendages such as hair follicles, and how a developing tissue senses and eliminates less fit cells by neighboring cells that are more fit. As I mentioned, skin tissue stratifies in mouse embryos between days 11.5 and 13.5 of development. These images show mouse embryos in this window of time. Looking carefully at the different layers of the skin, we can visualize the numbers and positions of cells in the lower basal layer and upper suprabasal layer. At 11.5 days, there are just a few suprabasal cells, marked here in yellow, sitting atop the basal layer, marked in blue. By 13.5 days, nearly the entire basal layer is covered by suprabasal cells. In order to understand how these different layers grow dynamically, my colleagues and I worked to develop a new technique to perform long-term time-lapse imaging of embryonic mice. The technique involves placing an embryo onto an air-permeable membrane that encompasses the bottom of a plastic dish. We then encase the embryo in a custom-designed 3D printed holder encased in a gel made of agarose, and then fill the dish with cell media. We found that when we use an air objective that allows air to permeate the dish and reach the embryo, the epidermis in contact with the membrane remains viable and proliferative for up to a day, enabling us to take videos of the cells whose specific parts, such as nuclei and membranes, we labeled with fluorescent markers. Here is an example of such a video, showing the growth and rearrangements of the epidermis over approximately 24 hours. The embryonic tissue is extremely dynamic. This type of imaging was among the first of its kind that enabled long-term imaging at this particular stage of embryonic development, offering us myriad insights into the growth and dynamics of this tissue. The videos enabled us to see what we call parallel divisions, which are when a cell in the basal layer divides and produces two daughter cells also in the basal layer. We observed perpendicular divisions as well, in which a cell in the basal layer divides and produces two daughter cells, one in the basal layer and the other above it. Through this type of division, the upper suprabasal layer becomes populated with skin cells, enabling the tissue to stratify. Our examination of hundreds of such divisions showed that we could predict, based on geometry, which cells were likely to divide in a parallel manner and which in a perpendicular manner. How dense the cells are packed locally in the basal layer is the determining factor. When cells are sparsely populated, a division is likely to be parallel. When cells are densely packed by other cells, the division is likely to be perpendicular. Our live imaging technique enabled this analysis because we could track the fate of cells after they divide. We also used this technique to explore how epidermal appendages, such as the hair follicle, form. The hair follicle starts as an aggregate of cells called a placode. It then grows downward to form a hair germ, which eventually becomes a full-fledged hair follicle. We performed live imaging of these hair placodes and found that within the placodes, nearly 100% of the divisions were perpendicular, whereas in the regions between follicles, called the interfollicular epidermis, about half of the cells divide in a parallel manner and the other half in a perpendicular manner. Finally, we applied our live imaging technique to study how epidermal cells that are endowed with stronger genetic fitness are capable of engulfing less fit neighboring cells. Here, a loser cell, labeled in green, becomes engulfed by its neighboring winner cells, labeled in magenta. To conclude, I wish to reiterate how grateful I am to the Arnold and Mabel Beckman Foundation for supporting my postdoctoral research on the dynamics of tissue development. My research during my PhD was carried out in a physics department, and my postdoc was transformative in teaching me genetics and developmental biology, enabling me to become an interdisciplinary biophysics researcher. The novel techniques I developed in my postdoc offered many new insights into how tissues grow during a critical stage of development, with rapid changes in cell motility, divisions, and morphogenesis. These changes are governed by many of the same pathways involved in cancer, so there are applications of this work to understanding both healthy development and disease. Thank you again to the Arnold and Mabel Beckman Foundation for supporting young researchers at this critical juncture of their careers.